Hi. Before the afternoon session starts, I just wanted to say a couple of sentences. Hi, I'm Mriganka Sir from MIT, and I wanted to express my sincere gratitude to the Autism Consortium for what it has done. As a basic scientist, and speaking on behalf of basic scientists in the Boston area and elsewhere, I want to place on record the fact that the Autism Consortium illuminated and opened a world for us. The world of autism through clinicians who we came to know, like Lenny, like Sarah, like Chris Walsh, geneticists like Chris and Mark Daly and others who made seminal discoveries, and that opened the way for us basic scientists to figure out what it is that genes do via the animal models that we work with. And so to make a long story short, we hope we have contributed to unraveling autism. You'll hear some of the consequences of that actually in the afternoon session. And we look forward to taking forward the, both the hope and the promise that the Autism Consortium started. I want to thank the people, visionary people who started the Autism Consortium, and in particular, Anne and Paul Marcus. So thank you very much. And at MIT, of course, the Autism Consortium has had a transformative effect. It has helped us start the Simon Center, which today funds and is actively involved with the research of about 50 faculty, 50 researchers who devote some portion of their work to autism. And we hope and expect that that will bear fruit in the years to come. So thank you.
Good afternoon. I'm Mustafa Sahin. I'm a neurobiologist and child neurologist at Boston Children's. My task this afternoon is to chair this panel on challenges and rewards of translational research in neurodevelopmental disorders. We heard some very impressive talks this morning about the advances in induced pluripotent stem cells and human genetics of autism. But I think at the end of the day, all of us are really focused on finding treatments and improving the lives of individuals and families affected with autism. So that's why we want to start off this afternoon. And I'd like to first ask the members of the panel to introduce themselves, and then we're going to start what we hope will be a frank and stimulating discussion on some of the opportunities and challenges in translational research. Randy?
Starting with me. I'm Randy Carpenter. I'm a physician that specializes in translational research or translational medicine. And for the last 15 years, I've been working on trying to develop novel therapeutics for individuals with autism and intellectual disability, including most recently at Seaside Therapeutics.
My name is Jim Doherty. I'm a neuroscientist, and I've also been working in pharmaceutical R&D, trying to come up with new therapies for central nervous system disorders, psychiatry, and autism spectrum, and other disease areas as well. Working currently as head of pharmacology for Sage Therapeutics, a small biotech here in Cambridge.
I'm Alan Crane. I'm one of the founders of the Autism Consortium. I'm also a parent of a son who has an Asperger's profile. And I'm a venture capitalist, and I focus on working with academics on starting biotechnology companies.
Yes, I could say ditto to that. Like Alan, I'm a co-founder on the Autism Consortium here, currently the president of the Autism Consortium. Also have a son who is on the spectrum. I'm an investor. We invest in, I work for Atlas Venture, I'm a partner there. We invest in very early stage companies, primarily in therapeutics, and trying to bring new therapeutics to market, and certainly some of those are in the CNS area.
Thank you. So we have quite a diverse panel in terms of experiences. So I'd like to open the discussion by asking, where did we come from and where are we today compared to, let's say, 8 years ago when the Autism Consortium started, or even maybe further, 15 years ago? Where is translational research in neurodevelopmental disorders today, Randy?
Well, I'm going to take advantage of the pre-discussion we had and an idea that Alan Crane proposed, which is, I think what's really interesting is to think about what questions that we're wrestling with today. So 15 years ago, when I would go out with Mark Baer and talk about how the study of a genetic disorder, Fragile X syndrome, might give you insights into therapeutics that could improve cognitive function, function in society, people generally at that time, the question they would ask is are you crazy? Basically that's the state we were at. You can't treat a genetically caused problem with brain development, and it's too late to really make anything happen. And they would typically follow up with, why don't you come back and talk to us when your antipsychotics have kicked in? So we were really at the stage, the first stage, I think, of scientific discoveries where there's disbelief or even ridicule of some of these ideas. And I think that the change today is that when we would go out and talk to people and say, we think that we can focus on a genetic insight to have a targeted therapeutic that may actually treat some of the underlying causes, at least in a subgroup of people with autism, that the questions are different. The questions are, how would you measure that? I mean, how would you measure that in a clinical trial? How would you enrich your trial so that people that were in the trial that you actually enrolled could improve on the endpoint that you're measuring? I mean, should you focus on social function or irritability or cognitive function? And then as people come a little bit farther along, they say, well, wait a minute, how would you get it approved by the FDA? And how would you bring this drug to market? And I think that, what we've learned today is setting the stage to ask some of these questions. And I think that's really a sign of huge progress, is that the questions are changing. And the things that people are wrestling with today are changing. And there are actually therapeutics that have been in development, that are currently in development, that are targeting potential underlying molecular abnormalities and treating those with the goal of improving the brain's function, improving the ability to learn, and improving function of individuals with, I think the goal where I've been focusing is on trying to help people become productive members of society that can live independently and contribute to society. And that's really where I think that we're going to be able to see some changes in the future.
Jim?
I think for me, one of the biggest things that's changed over the last 10 years or so is that we're living in the midst of a huge leap forward in fundamental understanding of the neurobiology across a lot of the disease areas we're interested in, including autism spectrum. I think we would each rapidly say there's an awful lot more to learn, but there has been a tremendous level of advance in genetic and molecular levels, even cellular levels of pathophysiology and disease. And as thinking about it from the perspective of moving drug discovery and development projects forward, one thing that's changed quite a lot since I joined pharma research 15 years ago is it is now a much more intentioned process of trying to link that into what will be measured in the clinic and trying to identify some endpoints that can be used in early clinical studies to really give enhanced confidence that a particular idea, a particular molecular target is really going to take us down the track to lead to a true therapeutic benefit. And I think it's back to this concept of being able to link multiple molecular targets into a cascade, whether that's a synaptic network or an interacting network of enzymes and proteins. But you get multiple potential molecular targets that could lead to a detectable benefit. And I think one of the things that's changed is people are much more actively looking for tests that can be run early in the clinical process to help triage the good ideas that are really going to lead to clinical benefit from those that have marginal or no effect.
I think, fundamentally what we have in neurodevelopmental disorders and autism today that we didn't have 10 years ago is we have hope. We have some prospect that there's something that we can think about doing that resembles what we do in other disease areas, what we do in oncology or, autoimmune diseases. 10 years ago, I was working for a big genomics company, Millennium, and we were always thinking about, how we were going to apply our advanced technology to different areas. And we engaged with different, clinicians in different areas. And 10 years ago, 10 to 15 years ago, it was hard for people to even conceive that you could have novel therapeutics that would fundamentally treat autism. I think that notion has totally changed given our deepening understanding, and I think there's even a notion that we may not only be able to affect the course of certain patients with autism in terms of their progression, but we may even be able to reverse some of that course as well. There's biological evidence to suggest that. So I think we're still very early, but I think fundamentally we have that hope, we have that concept now that we can make a difference. And by the way, that's the first thing that you need for people to systematically be able to focus on it and make it happen. Because it's early, though, we're going to see fits and starts. So, several years ago, and I don't know if there are people from the pharmaceutical industry here today, but several years ago, there was a lot of enthusiasm among pharmaceutical companies, among the big pharmaceutical companies. In fact, we had a whole special event around the Autism Consortium, with those companies, and there have been a couple of visible failures in pharmaceutical companies in this area. And so I think people have backed off a little. And so it's still early. I mean, we're going to see fits and starts. But I think we have to keep, fundamentally pushing forward. And one of the other concepts we discussed sort of on our pre-call, getting ready for this, was that we now have challenges that we didn't have before. And that's an optimistic thing because we've advanced to the point now where we've made enough progress so we can see what the challenges are going to be in developing therapeutics. And we'll come back to that, I think, as we progress.
Yeah.
So I'll agree with all you guys. But being at the end of the line here, let me just add, sort of from an investor perspective, how we might look at it. Our job in venture capital is to take risk. And in some ways, the pharmaceutical companies moving out of this space, or at least backing away temporarily from this space, gives us an opportunity maybe to take more risks and make investments that will develop new therapeutics in the space. But as we look at any investment, there has to be a hypothesis behind it of measured risk, that we at least understand what some of those risks are and how we would overcome them in terms of studying a particular biological hypothesis or chemical modality hypothesis to proceed. I was recently at a conference where people were reminiscing about progress in cancer, and someone was reflecting on the days when IRBs would not allow biopsies to occur because they were afraid that the cancer would leak inside this person. And they've obviously come a long way from utilizing the information that is resident in a tumor to actually molecularly target tumors. Now, we're a long way from doing that in the brain, but we're starting to get tools, if you will, that allow us to have handles on testing the hypotheses that we make. And good examples are not only what we were talking about in the clinic, but models. And the ability to create, as you saw earlier, IPS models that would help us interrogate the disease biology and be able to intervene at least in a in vivo, if not in vitro system, to understand the disease biology better. Alzheimer's, a neurodegenerative disease, has really been a challenge in terms of understanding that, not in humans. And just recently, this past week, Rudy Tanzi published the paper that showed a more relevant disease model that actually showed the tangles that are involved in Alzheimer's and their ability to now test that in vitro. So these kind of milestones give us a lot of encouragement that we will have tools in place. It is unheard of to actually provide multiple spinal taps on a person during, a CNS drug discovery. But that information in the CNS fluid and better ways to do that are really important. So what all of us are looking for is better ways to test these hypotheses, first, if we can, in animal models, but more importantly, related directly to outcomes. So whether it be things like what's happening in the cerebral spinal fluid, or what we can learn from imaging techniques about what's happening in the brain under therapeutic intervention, are key things that are things we're starting to get in hand and give us hope that there will be more rapid progress in the future.
Great. Before I go to the next question, I just want to add my clinical perspective, if I may. Compared to 2002, when I started the tuberous sclerosis clinic at Boston Children's, I think we've come a significant way in at least taking care of patients with tuberous sclerosis. So for those of you who are not familiar with this disease, it's a genetic disease, and half of the patients with tuberous sclerosis are affected with autism spectrum disorder. Back then, we were actually quite poor in diagnosing children affected with tuberous sclerosis with autism. If you were diagnosed with tuberous sclerosis, people sort of assumed they had only tuberous sclerosis, and autism diagnosis was not being made. And in fact, many institutes at the NIH would not fund studies on tuberous sclerosis, even though half of the patients with that disease were affected with autism. So it wasn't recognized as a bona fide cause of autism back then. Now, going forward 12 years, we have about 200 patients in our clinic. Essentially, every patient that walks in through the door is part of some trial, either genetics, imaging, or biomarkers, or even treatment trial in tuberous sclerosis ongoing in our clinic. And then NIH has been very generous in funding some trials looking for biomarkers in this disease. And we've been in the midst of a phase 2 trial treating or trying to attempt to treat tuberous sclerosis with a small molecule. So from a clinician perspective, things have moved a huge way. So I want to turn to each of you, and some of you already raised this, is now that we've come this far, what are the challenges that we're having right now? What are the pitfalls in the trajectory of the translational research that has been moving forward in the last decade? Randy?
I think the challenge is we need to have one success or maybe a couple of successes. We need to show that this is a fruitful path forward and that it's possible to deliver on the promise and that we can develop treatments that'll actually make a difference in people's lives. And I think once that happens, then the investments will follow. And so it just need to figure out how to do it. And what we did in our company is we looked out and said, who are all the key stakeholders and how can we put together, an effort to make this happen? So we started with expert clinicians and physicians. We talked to parents. We worked with advocacy groups. We interacted with the FDA. We put in collaborative and cooperative agreements for development with NIH. And each of these groups have to learn from each other. Many physicians had no idea what would be required for regulatory approval of a drug, and basic scientists really didn't understand the process. So getting these groups to talk together, I think, has really opened up the door. But I think that what we need to do now is execute on this promise and deliver, and then I think that's the key next step.
I would absolutely agree, Randy. I would add to that, I think, again, thinking about some of the presentations this morning, for me, a lot of this is on the research side, to start to understand some of these de novo mutations in context. So there are quite a few different genes that seem to lead, deficits that seem to lead to overlapping or similar pathological effects. And so which ones are the ones that are leading to systems that are places where one can make an intervention? And so that's I think the research challenge on the clinical side of things. Really, it's a stepwise progression to get, the ultimate goal is to get to a clinical trial where you can measure endpoints that will demonstrate improved living, improved function, and hopefully be approvable by the FDA. But even before getting to those steps, there are several smaller steps in the process of being able to demonstrate that a particular therapeutic, a particular drug candidate is getting into the brain when someone takes the pill, or that it is interacting with a biologically relevant process that gives you some confidence to take that next larger step. And it's really developing those biomarkers, whether they be biomarkers for brain exposure or ultimately for efficacy, that are key steps along the chain to getting some novel therapies all the way to the market and to the patients that need them.
I guess I would echo what Randy and Jim said. I think speaking as an investor in biotechnology, in general, CNS disorders are hard to invest in, and that's because often the proof of concept comes in a large phase 3 study in people, which means there's a massive investment that has to occur, which means you're beholden to some major pharmaceutical company to be able to realize your product, which means you can't cut a good deal with the pharmaceutical company, you can't create a lot of value, which means people won't invest in the company in the first place. So it's kind of a vicious circle, and it really makes it very difficult. And it's made even more difficult by the fact that the endpoints in many CNS disorders are just not as precise as endpoints like we have in other diseases. Like, in oncology, we look at overall survival, we look at RECIST criteria for the shrinkage of a tumor. They're very quantitative, very specific measures. I think that it's further compounded when you come into disorders that haven't had established therapies. The only approved drug, I believe, still for autism is Risperdal. Is that true, Lenny? Yes.
Abilify also.
Yeah.
Abilify is as well now. Okay, so
Two.
yeah, so there's two. They're in the same class and they treat symptoms. So I think that we don't have the regulatory pathway, we don't have the clinical pathway, we don't have the endpoints, and I think this is something I'm sure Randy will come back and talk about more later. And Jim brings up the issue of biomarkers. I think biomarkers are really revolutionizing the way that we approach clinical development in other areas. For example, in oncology, we can get so much more mechanistic insight in people relatively early in clinical trials, even in phase 1 studies and certainly early in phase 2 studies. And I think that they're absolutely going to be transformative for CNS disease, in particular for autism. Many pharmaceutical companies are now starting to build big translational groups that are focused on biomarkers in the CNS. So the mechanistic insight, a lot of what we've been talking about here today, the emerging mechanistic insight, those will be our drug targets that we want to go after to understand therapy, but they're also going to be the basis, that mechanistic understanding is also going to be the basis of understanding the biomarkers so that early in the clinic we can start to understand, as Jim said, are we getting the drug in? Pharmacodynamically, is it having an effect? Can we start to appreciate the value of therapeutics earlier? And once we do that, I think it reverses the cycle and we can really start to get people engaged, and really the biotech community, the venture community, the pharma community will get much more engaged in moving novel therapeutics forward. So I think we're on our way, but I think those are fundamental challenges. I would love to be able to invest in autism drug companies. Right now, that would be, I think, almost impossible, to do, given where we are. But I think we're on the way there, and hopefully in the next, certainly I hope it doesn't take 10 more years, but I hope in the next few years we'll be in a position where that becomes more possible.
Well, just let me add briefly to that, is that the other issue is, we all know, is what is autism is pretty broad-based, it's pretty heterogeneous set of syndromes. And even when we have a particular drug with biomarkers, what we will do is try to take a lesson from other diseases where we can do a small subset of the patients that are more homogeneous. So even if it is only maybe 1% of all autisms in this particular trial that we're going to go forward with, understanding how that drug works in those individuals, and obviously if it works, it's great, but then understanding what are the overlapping pathways from that particular condition with the other autisms, if you will. So I think that's a lesson that we've learned in a lot of other diseases, rather than doing as Alan was saying, the really big trials, which are costly. We try to refine the number of variables, reduce the number of variables, and the patient, of course, is a variable here, so that we can study these things effectively and extrapolate from there where these drugs, either mechanistically or from different indications, patients may go. So that's a very important thing that we do here in other indications, but have to translate that. And certainly things like tuberous sclerosis and Rett's are ones that are doing that. But once we understand the underlying mechanistic of the other forms of autism, we'll be able to do that more intelligently as well.
I wonder if I can follow up with Jim. One of the issues that comes up when you go to talk to pharmaceutical companies or even to the NIH is that some of the results that had made headlines from the academic work seem to be not necessarily that rigorous or reproducible when they are tested in other settings. And I'm wondering how much of a problem that is, and how much of a problem, both sociologically, obviously, in terms of what the science community stands for, but also how much of a real problem it is in terms of drug development.
It's definitely a variable challenge. I mean, that is something that that people look at very, very carefully when exciting new results come up. Is making sure that before making a large investment in time and resources that's an observation that will hold, or a path that is really going to drive towards clinical utility. So it's definitely a question that gets asked all the time.
Yeah, just to add to that, I mean, it is I don't know, to say it's a problem, it's an issue, right? You don't want to really be so negative on academic work, but as part of when we do investments, and Alan just the same, is we do what we call seed investments, and very often we're translating academic research into a company. But the first thing we need to do is to reproduce that data in a much more rigorous way or robust way than is actually possible in an academic setting, with many more samples, with more controls, and with firms that do those kind of assays every day. So it is a very important part of the process of investing in this space or understanding this space is that a recapitulation of the data that comes from the academic settings and understanding it and utilizing it effectively. So,
are there other mechanisms that could be put in place that would sort of encourage that make that part of sort of scientific practice? Because it seems like that could potentially accelerate things, not slow things down, but actually accelerate things to be able to validate certain results. I know there are various foundations, et cetera, that have been looking into this, but I was wondering.
In my view, absolutely. I mean, part of the issue actually is not so much the original work. It's that it's a slightly different question often that you're asking in a translational sense to develop a therapy. You're asking specific questions around, will this particular approach work at a given level? How much of a particular molecule needs to get into the brain, and for how long, to have a therapeutically relevant effect? And these are questions that are central to pharmaceutical translation that are often not central to the original question of defining the model or defining the role of the particular mechanism in the disorder. So I do see an opportunity for collaboration in asking slightly different questions that would then facilitate the development process.
I know that when we're involved in working with academics on some new breakthrough and building a company around it, often it's really important that the academic has sort of engaged in the process of beginning to think about translation and working with people in the field of translation, even as they're doing work in their academic lab. And the best kinds of science to build a company around are science that really is starting to try and answer some of those translational questions. Now, it's not going to be as robust as the CROs that Peter described, but to ask the question in lots of different ways, and if it's a cancer therapy, try it on lots of different tumors, ask lots of different questions, and really, the single experiment that you base a company on is much more likely not to reproduce than something that's been tried by multiple investigators in different ways and where you've started to answer those translational questions. So part of it does go back to the academic lab and even how you can do your research so that it's more likely to robustly result in something that moves forward. Because the last thing you want to do is do years of research in your lab and then have it, start a biotech company that gets seed funded and then have it, quickly, fall on its face because some key questions weren't really asked.
But I think, from our perspective, we've taken huge leaps of faith on science and turned them into companies if the science was so compelling in its innovation that it would have made a material clinical difference. And we're actually in the midst of a couple of those now where the observations are sort of something that biologically is important, is upregulated, where actually the therapeutic may not be well known at this point, but at least the biology is so interesting and could make such a difference in a particular disease indication that we'd be willing to marshal that around. And sometimes it means still continuing to funding research in the academic lab to further the understanding of the disease biology, which, as you probably know better than I, gets very difficult in the Harvard system, especially if people are involved with companies. But it is something that we're certainly not afraid to do with science that's really innovative.
Maybe I can follow up on that and sort of ask you, each of you, to imagine what would be the ideal translational team that might be required to address some of those really challenging scientific questions that we have to address now. I think, as Matt State suggested, there's been huge amount of progress in gene finding, but in some ways that's a problem that we had the tools to answer potentially, whereas we don't necessarily have the tools to address the other questions that we're starting to struggle with, like the circuitry questions, or to be able to look at human tissue for the first time in culture kind of questions that are, the technology is just coming on board. But what would you imagine is necessary if you were to sort of come up with an ideal translational team? How would that be composed of, and how would it function to be able to get to these really hard questions in terms of finding functionally successful interventions in autism? Randy, do you want to take a stab at that?
I get to start with the easy ones, right? So I think there's really two parts of it. One is, what's the scientific team that actually makes you believe that it's a potentially translational or transformational scientific discovery? And that's largely often in academics or in these early guerrilla stage startups. I think the part about translational beyond that, of opening an IND and doing clinical trials, is probably less interesting. But to me, the challenge today is the broad set of skills that need to collaborate because of the tools that we have available. The amount of data you generate when you genotype an individual, interpreting those data, are the reads accurate? What do they mean? What pathways do they project upon? And integrating people with informatics skills, biology skills, scientific insights. You really need a multidisciplinary team that can look at it from many different aspects to sort out whether or not that this is really something that we believe could be effective as a therapy. And it's just getting harder as we have more tools and generate more information. So, and then there's the chemistry within that team also. You need people that respect each other's expertise and discipline. So this is partly, you can pick the different parts, but part of it's also an art form to get a highly functioning team. And I think that's a big challenge as we go forward, because there's not a single person that knows enough about every area to do it all by themselves. So progress today requires intense teamwork.
I think that's actually a key observation. That I would agree entirely that this is a complex exercise in really trying to move from a deep understanding of complicated biology into something that is measurable in a clinical environment. And there are multiple challenges involved in that, and I think it's more too much to expect that any one individual is really going to carry enough expertise to bring all those pieces. So it really does mean pulling a team together, and I'll go back to this concept of understanding the system's biology and really how to put targets in context to the system, and then from the clinical side of things and from the measurable side of things to be able to identify those biomarkers or those modalities that will allow you to measure whether you've perturbed that system biology. And then I would agree about chemistry, and the other piece I'll add in is something I mentioned previously is pharmacokinetics. So understanding how the potential therapeutic is handled by the body, how much gets into the brain, how is it metabolized, how long does it stay in the brain. So it's a really complicated process, I think, is the bottom line, and it takes I think multiple minds to really handle a translational project for any target.
Again, I can give a real-life example. Unfortunately, it's not from CNS, it's from oncology, but one of the companies that I recently started is focused on the tumor microenvironment, some very novel mechanisms around oncology, and founded out of MIT and MGH. So our academic founders are very actively involved in the company. But in addition, we hired as our CSO someone who both has a tremendous amount of expertise advancing products into the clinic, but also has a tremendous amount of expertise in biomarker development. And sort of that going hand in hand, adds insight as you're moving your products forward through discovery and into development. And then we have an expert in animal model systems, because each of the questions you're trying to answer is different. And by the way, it's easier to answer questions in animal models in oncology, but our animal models in oncology aren't even so good. And when you're trying to modulate the tumor microenvironment and not just kill the tumor cell, you really have to ask the questions in a sophisticated way. And you have to do that in every area. And then I would agree that having specific preclinical and clinical expertise that can answer the PK questions. And then there's the drug side of it, which is you have to have an expert in the process development of making your drug and making it reproducibly and analyzing it and making sure that what you are using is consistent and then can be brought forward into formal IND enabling studies and ultimately into people. So it's a very multidisciplinary process, I would say that there's many special aspects that would have to go into advancing autism drugs in that way.
Before Peter gets a chance, I just want to say what you're describing is obviously multidisciplinary, different skill sets, and it's most likely not present in one institution or in one lab or one company. So that would require sort of lowering the boundaries and having collaborations between multiple institutions, between academia and industry, or within academia, between different departments and divisions, it sounds like. And
yeah, I mean, we assemble those capabilities, within the companies.
Within the company.
But if we didn't, we need to work very closely with the academics and really bridge all those disciplines together. And really the greatest strength comes from that whole team, including, your academic founders actively, engaged in the process as well.
Yeah, really not much to add, but I think it really depends upon the program you're working on, what you have to assemble. So there isn't sort of the ideal dream team of translational scientists or scientific team. In some cases, you may know the biomarkers. In other cases, your most important problem is to create those biomarkers. And so you need probably quite a different team in those two, just as in that example in itself. And when we start even these small companies, we do leverage tremendously outside. I mean, the amount of CROs that we can use in a small company that's going into a clinical trial can be 20 different firms or consultants that are helping progress that through the clinic at any one time. And that's why it is such a difficult thing, what we're doing. So it is collaboration clearly with the academics, clearly with just CROs, and then specialized consultants in particular areas, or specialized people who can develop particular assays that really help us understand target engagement or biomarker discovery, biomarker measurement. So there's no one size fits all, unfortunately, and that's why I think in some ways, what you're trying to do with children's, Mustafa, is hard, because you need a lot of capabilities, and I'm sure just like other parts of the hospital, there may be a little financial pressure here and there, and you may not be getting all the resources that you would desire.
But I think Peter's referring to the Translational Neuroscience Center we started about 18 months ago, and one of the first things we learned, as Jim was pointing out, is that we are not very well trained in PK and PD. Pharmacokinetics and pharmacodynamics are things we really don't think about when we're publishing papers in neuroscience or cell biology, to be honest. And those matter hugely in terms of drug development. And so we've actually decided to hire people with pharma experience into children's to be able to help with those. But at some point, it's very clear that we're going to have to work and leverage with pharma at the same time. But those are the kind of, I think, collaborations that are going to be absolutely necessary, at least from my perspective as a translation researcher, to move things forward. We spent quite a bit of time talking about the sort of scientific barriers, scientific challenges, like heterogeneity of autism, lack of objective outcome measures, lack of predictive biomarkers. I was wondering if we could spend some time talking about the sort of structural, systemic challenges, Randy. What are some of the issues that, if we could solve those, could make the research go faster?
I think that one of the biggest problems we have today is we have 21st century tools and assays and capabilities that are still being constrained by 20th century drug development approaches and regulatory approaches. So what do I mean by that? I mean, we heard today about individuality, people with the same DSM diagnosis of autism having perhaps 1,000 different genetic causes and highly variable symptoms. And yet when we do clinical trials or we do development, we try to have a uniform population-based treatment that has uniform effects and needs to work in the full population. And I think we should be having our eyes open and looking at what's going on in other fields. So for example, in cancer today, they're looking at trials of super responders. So maybe only 3, 5, 10% of the people in the trial had an unexpected, very, very big response, or other people have no response at all. And looking at those and really gaining insights. And it's actually possible to figure out ways to get drugs to market that aren't working in everybody. Even in genetically defined populations of tumors, the drugs don't work in every person. And that's not very surprising. I think we took this approach of looking at Fragile X syndrome because it's a single gene that's mutated and you don't make a single protein. And we thought this will get us a much more homogeneous population. And in fact, in our trials of over about 450 people with Fragile X, if you're a male and you have this full mutation, every single child was on the autism spectrum, and about 3/4 met full diagnostic criteria for autism, which is a bigger percentage than what you see in autism spectrum disorders in general today. And yet, but they're not phenocopies of each other. There's wide variability in these males in the amount of severity, the symptoms they have. And that shouldn't be surprising, because if you take the Fragile X mutation and you put it into 7 different mouse strains, you get 7 different phenotypes. And so the people have sort of ignored the background genetics, environmental factors, parenting, whatever, that lead to differences with the same mutation in the same person. So if you actually correct the underlying molecular pathway, or you modulate it and improve it, you'd expect different behavioral effects in different people. And yet, although we saw improvement in global measures in our trials, we didn't see improvements on any single specific endpoint that would reach the current requirements going forward. So I think there needs to be a rethinking about this, and I would encourage you to look out at some of the solutions. There's some radical ones like free to choose medicine, which are proposing an alternative pathway to the FDA altogether. And there's other groups like the Manhattan Institute, this Cure in the Code, published by Peter Huber, is a very good book that talks about this exact conflict. And I do think that there needs to be education, there needs to be some movement here if we're hoping to see something go forward, because a drug that is very, very helpful, or a treatment that's very, very helpful in 25% of people with autism and or intellectual disability would be really a major advance. But it's very difficult to figure out a way that you could actually get a drug like that to market today when we can't prospectively predict who's going to respond. We just don't know enough right now. So I think a major systemic thing is trying to advance people's thinkings about what a success would be. And my personal belief is it's time to start focusing not on reduction in aberrant behaviors that may reach statistical significance, but improvements in function. And I do think that that's possible with some of these treatments. And one treatment's not going to work for everybody, but I think we need to rethink the whole development process.
Anybody else?
Yeah, well, I would just echo one of Randy's key points around being able to select patients that are likely to respond to a particular mechanism in early trials. I think it's, when you consider that any given idea has a certain lifetime, if there is a failure early on in the lifetime of that program, even if it's the case that there are other patient populations that might have responded, if there's no way to know that's the case, you'll never get to find out. So really, I think that you have to be very careful about identifying the patient populations that you would like to test, especially very early on in the process of testing an idea. I think if you can get some positive results that would suggest that you've got a handle on a therapy that's likely to be of benefit, you can start branching out and asking the question, would other subsets of patients respond? But you often don't get that chance. And so really an intention to look very early on in making sure that it's the right patient populations that have the greatest chance of responding, probably using biomarkers or functional endpoints to identify who those people might be. But I do think that's a key opportunity and challenge moving forward.
So can I push on that point a little bit more? So we all talked about biomarkers several times. What, from your perspective, are sort of the most encouraging biomarkers right now that will sort of open up that question to predict who's going to respond?
Well, I would echo something that Peter said a few minutes ago. It's not my view that there is any single biomarker that is likely to be beneficial across the board, and I think that's one of the challenges is to sort of link the appropriate hypothesis to an endpoint that's likely to detect that. I do think that a number of functional biomarkers show an awful lot of promise. I was very interested in the talk given earlier today around epileptiform activity. That's the EEG and other forms of evoked potentials are relatively easy to measure. As we heard about today, that the existing technology is not as good as it could be, but at the same time, a lot of that technology in parallel is evolving very, very rapidly. So I think you could see a time in the not very distant future where those electrophysiological techniques are readily accessible. They also have the benefit of being cheap and portable. But certainly imaging modalities can be critically beneficial in understanding what's happening in certain parts of the brain under certain conditions, and also can be used to tremendous value. And I would also say there are other examples where biochemical biomarkers can be equally powerful. So it really is a process of linking the right biomarker to the right hypothesis.
I think we are all focusing on biomarkers just because measuring outcomes otherwise in this patient population has been extremely difficult, whether it be cognition, behavior, that those measurement technologies or ability to measure them in an objective way are very difficult. So I think that's an opportunity on one hand, is to have better ways of measuring those. As you know, we supported the part of the Rett trial at Children's, and what got us interested in that was, gee, you can measure heart rate or respiratory rate, autonomic functions that are totally objective. That there is a system that everybody will agree is the measurement there. And yet now the trial is going forward in anxiety because that's perceived as what the FDA will be more favorable to sort of a proof of concept in a trial, a phase 2 trial in Rett syndrome. But there's genetically defined group with something that's very measurable. And now my concern is anxiety, of course, is a more subjective measurement. And so I think that's the big challenge. And that's why you'll hear everybody in this field talk about biomarkers, and why the Autism Consortium actually helped facilitate with Simons the biomarker project is to try to move that along.
Right. I mean, if we had good quantitative, physiological measures, like eye movement and things like that are very reproducible, and we can get, therefore, good early human, signals, I think that would be very important. One other point I wanted to come back to on sort of the reductionist view and, 1,000 different genes, 1,000 different autisms. It becomes daunting. I mean, from a drug development standpoint, we're not going to make drugs to every one of those 1,000 autisms. It's just not feasible. So it's also important that we're thinking about what are the common pathways, what's upstream, how do these genes, not only what are the genes, but how do they relate to each other, and how can we begin to, it's fine if we had 10 different drugs to treat different forms of autism, but 1,000, is not practical. So it's really important for us to understand that interrelationship. And hopefully we'll start to get some insights into even how some of these are interrelated in terms of the phenotype as well.
Yeah, that seems to be a critical question. How are we going to find the convergence among the 400 to 1,000 genes that Matt State mentioned? And he proposed, I think, one of the ways would be sort of gene expression in terms of spatial and temporal distribution of the genes, etc. But are there other ways that people imagine these genetic causes could be stratified, or common pathways could be found among those causes?
Well, the approach we were pursuing is this pathophysiologic axis around regulation of protein synthesis at the connection between neurons, with an axis between your area, TSC, and Fragile X, one leading to overactivation of this pathway and one under. And you can actually define a proteomic profile that defines activity along that axis. And the question is it something that you could actually measure in peripheral blood? You can certainly see it in the brain, and we never got to finish answering that question. But I can imagine if you have a regulatory pathway that's controlling gene expression, protein synthesis, that you could have a blood test that would say this person has overactivation of this pathway and this one has. And on this axis, it could be that it looks like it may be relevant for half the people with idiopathic autism. It's still early days. So that does, the limited data that we've seen, and whether you start from the bottom up with a single gene, highly penetrant mutations that are associated with autism, or you start at the top down with autism of unknown causes, can we understand anything about the genetics? You end up converging in similar pathways. And that gives me hope that there may be 1,000 different genes, but they're going to converge on a manageable number of pathways. And you're either a lumper or a splitter, and I tend to be more of a lumper by nature. So my hope is that we will have a limited arsenal of therapeutics, and they won't stand alone. It's not just that you're going to improve signaling in the brain and plasticity of the brain, the ability of the brain to learn from experience, but you're going to combine that with therapy or school or training. And so I think we need to think holistically across the whole arena here. And again, my focus has been on the more disabling intellectual disability and autism type of an approach, because I think we've had more insight into highly penetrant single gene mutations that give us more insight into the biology there. And so that's where I've been focusing, and my hope is that some of these medications with apps also help be more broadly applicable. And I would say our experience to date is consistent with that.
I agree with you, and I'm hoping you're right, that you're all right, that these 100 to 1,000 genes will converge on a manageable number of pathways. Otherwise, we're in big trouble. I think that we're at a point, though, where we can start to test those possibilities. At least my group feels like we've come a certain way with testing mTOR inhibitors in mouse models and cell culture and patients with tuberous sclerosis. Now we can go to the next closest gene, which is P10 from our perspective, which should respond somewhat similarly. So we actually have a project with the NIH to do mTOR inhibitor therapy trial in P10 patients. So I think what's exciting to me is that now we've gone from one disease at a time to hopefully comparative pathophysiology of more than one disease, where we'll see if those pathways are truly common or not. And I think it'll be helpful either way, knowing whether a drug helps two genetic disorders or it doesn't help the other one. I think is really important for us to know at this point.
Data's good.
So we had started from talking about how far we've come in autism translational research, and we spent quite a bit of time talking about some of the nitty-gritty challenges in this field, and some of them are quite large, but I just want to sort of, before we turn to the audience and ask for their input, ask each of you to sort of summarize some of the solutions you are seeing moving forward to address some of these challenges. And if we were to have this discussion in, let's say, 5 years, where do you think we'll be in that time? What are some of the steps forward we'll have in the next few years? Randy?
I suggest we give Peter a chance to talk first
Oh, right.
this time, so then I can just reaffirm what everybody else says.
Oh, you caught me sleeping too. I knew there was at least 3 other people who were going to say something before me. Well, I think we've talked a lot about, what are the things we need. I think 5 years from now, I think the convergence of that understanding, both on the disease biology side and how to really interrogate it through the process of drug discovery into the clinic, is going to be an important step. And so my take is we will see multiple programs in different autisms, if you will, in the clinic in 5 years. And some of those will fail, but certainly some of those will give us either a pathway to a new therapeutic, or at least the better understanding of the disease biology, so that we can develop better intervention points. And it's going to take all the things that we've been talking about, the reductionist of the genes that we're looking at into sort of pathways, the better understanding of which of those intervention points are good, biomarkers that allow us to measure the effectiveness of those, and frankly getting these things into people, because ultimately that's the only model that really matters in this business.
I think the solution sits here in this room, or it's certainly symbolized by us all sitting here together in this room. I think it is the convergence of clinicians, scientists, patients, families, caretakers, all together. We're never going to have a solution without that. I mean, that's going to give us the clinical, studies that we need. It's going to support the development of mechanistic insights and biomarkers and variation within autism. I think all the elements that we're trying to get at in terms of better biomarkers and all that is not possible without that continued intersection. So I hope that this, continues to be a very, robust community, and that, hopefully other communities around the country will be inspired to, work together in similar ways. I agree absolutely with Peter in terms of 5 years from now. I think we will have a number of interesting, products in the clinic, and I think we'll start to see some success, and I think that will just beget more success. And, we'll start to really get to a place where there's a growing level of excitement. Obviously, there's massive unmet medical need in the autism area. The idea of a therapeutic that could slow the course or reverse the course of disease would be absolutely huge. And I think people will start to get excited about that potential as they begin to see scientific progress early, but in the clinic.
Yeah, I expect that in 5 years we'll be debating the relative merits of some approaches that people are trying. I think there's no clarity today about which of these modalities or which of these metrics are going to be more impactful than others, and we won't know until we find out. But I do think that there has been a dramatic change really in the understanding or the appreciation of the value of what we're calling translational neuroscience. And I think we've gone beyond the point where you're in the position of trying to convince organizations that this is really important to do, to now everyone rolling up their sleeves and going through the complicated process of figuring out which are the best approaches. So that's what I expect in a few years. We'll be debating the progress and which ones are going to be most impactful.
Back to you, Randy.
Yeah, I think that...
You're supposed to agree with the rest of us first.
So first of all, I agree with what everybody said, and I completely do. And I actually was... I was thinking, oh gosh, I can't say that one now, but no, but I think that one of the things that I think is really important is we're at the very, very early stages of trying to figure out how to actually do clinical trials in individuals with autism. If you look back on a review article from 2010, which is the last time it was summarized, over the previous 30 years, in the field of autism, there had been a little over, somewhere around 40, between 40 and 50 pharmaceutical drug trials that were placebo-controlled and randomized, so a way that you could actually interpret them. Over 30 of them had only a handful of people in the study, and only 2 had 100 or more. Only 2 trials had ever been performed with 100 people in the study. That's where we're working from, and that's where we're looking at. And the only real success ever was to give a major tranquilizer, an antipsychotic, that to children, then reduce their irritability. So we only had one measure that could actually detect an effect. And I think the way for this to go forward is the parents and the community need to be more engaged and involved in research. We need more information about the natural history, the diversity. I would encourage people to have DNA drawn and actually get tested so you have some good phenotypic, so some good behavior to correlate with the genetics, which is often lacking. You get genetic samples, but maybe you haven't really characterized the people well. To participate and engage in being part of the solution going forward. And I'm not encouraging people to be in trials with brand new compounds of unknown safety. I don't think we know enough about how to do the trials yet. Personally, I don't necessarily recommend that. But compounds that we know are very, very safe, that have been in millions of people, that we know we're not going to harm someone, we need to do those studies to find out. Can you actually give these tests? Can they measure anything? Are they reproducible if you do them more than once? Can we have better endpoints? Can we figure out how to do the studies? We just don't know enough. There's too much that's unknown right now. And so I think that I would agree with Alan. Part of the solution is in this room, is be part of this, be part of the solution, and participate in the Simons Foundation's genetic sequencing trials. Participate in other trials that are just providing information about what your needs are. What are the problems you wrestle with every day? What are the things that are important to you? And that's what I would encourage people to do, because we need a lot more information. And I see a future where the insights and the knowledge comes out of trials in humans. Why do these 30% of the people hit their individual education goals at school this semester for the first time, and the other 70% don't? Can we understand why that happens? Those are the types of things I think we want to have. Real meaningful functional improvements that matter is where we should be looking. And so that's what I would ask.
Thank you. I also want to thank the audience for being very patient for the past hour as we were going back and forth. But I think this is the right time that I want to turn to the audience. There are lots of different stakeholders in the audience, and I would love to welcome any comments or questions on what we've talked about. Luke?
Barrett, be curious about the panel's reaction to there are some features of autism compared to sleep, compared to GI functions, which they really are quite quantitative data. At least some of them don't even involve the drug penetration of the CNS. And I was curious about, these are all things that really make quality of life better for both individuals and families. And what you saw as the barriers or potential for getting at least some focus on what I'd say perhaps more practical features.
Everybody hear the question?
Maybe if we could repeat.
The question was, in the GI, there is obviously effects in the GI that are quite measurable, but also linked to, autistic conditions, and what is the panel's view on that? Is that the short version of what you? And translational level. Yeah, actually, we as a firm have actually been following the microbiome space now for a couple of years and trying to figure out where is the best place to intervene in that and understand it. And I think it is just coming, the tools, again, of being able to measure, disease versus non-disease or conditions are really becoming of age, if you will. And I think there's real opportunity there. How to approach it, though, from a therapeutic standpoint, whether it be the microbiome replacement or the approach we're taking actually is the combination of engineering synthetic microbes to actually go in and sense a condition and create a therapeutic response is a little bit Star Wars, but it's kind of the things we do sometimes. So I think it's an excellent point that you make, is that could be more near term, something measurable, and has sort of built in its own biomarkers, if you will, if you can measure it.
I mean, the other part of that is, of course, well, obviously the gut that bacteria, they also have neural nets, presumably it's functions in the brain by the same molecules
Yes.
that function in the brain. And, you know, these are amenable to drugs that don't have the data.
Right.
So I would just like to emphasize one of the points you brought up, though, which is the sleep issue. And I would say that there are, it's not as well studied, but there are certainly a lot of these genetic mutations that cause circadian disruption and abnormal sleep-wake cycle. And I know a lot of parents have children that don't necessarily sleep every night. And that's a problem not only for the individual with autism, because we all know that sleep deprivation impairs your ability to learn and affects your mood. But it's also a problem for the parents who are often up at night and not able to sleep, and big effects on quality of life. And I do think that there are some good approaches and some possibilities for drugs that would improve sleep that could have dramatic effects on daily function if you sleep better at night, and on family dynamics. One of the problems, though, when you think about drug development is this issue of pseudospecificity type thing. And so you'd have to somehow show why it's unique, the sleep effects are unique to people with autism versus the general population. And maybe that's what's holding people back. And I think in the sleep field in general with new therapeutics, you already have generic drugs that are pretty cheap and pretty effective that are used as sleep medications. So it would take some brainstorming, but I do think I've been thinking about that a lot. I think that's a really important area that we should focus on.
Just to add to it, we made that one of the secondary outcome measures for our mTOR inhibitor trial in TSC patients. It's a huge problem in the clinic, and we'll see if that affects.
Hi. So I'm an entrepreneur that was an engineer, and I just want to follow up on the commonality thing, because that's how I got into this, about looking at one way to answer that question about how do you look the other way is I looked at sensory processing disorders. Common in that is deep pressure. And I think that's a way that you can start diving in, is deep pressure is a neurological stimulus to the brain, and then it's linked to oxytocin release or firm pressure and touches. So I think exploring the common thread of sensory issues to deep pressure, and that's common across a majority, or at least used commonly in treatment of children with autism, as high as 80% in certain age groups. So looking at that from a top-down approach, looking at those common threads and then figuring out how to link it back to the genes or the neurological expression, I think is really interesting and where I would like to go. But I'd also like to ask a question, probably more to Alan and Peter. I've gone to enough of these conferences. I spun out a medical device company for a sensory-based technology as an engineer, but all these conferences tend to talk about only drugs as translational things. And what are you seeing for devices and other technologies that aren't classic therapeutic drugs? And what does that market look at like, or issues, high-level issues that you're seeing, because that usually lags therapeutics, classic therapeutics?
I think the most interesting device opportunities are when there are sort of ancillary markets for devices, and some of those really don't, they represent very incremental improvements, but there are a number of fields where devices are starting to, potentially take the role of a drug. So, there are companies that are using, magnetic stimulation for depression quite successfully in patients who are refractory to, drug therapy. There are some device-oriented approaches to oncology now that involve, transmission of radio frequencies. There are actually device-oriented approaches to asthma that involve sort of remodeling the surface of the bronchial tubes and treating severe asthma. So I think where the device, there's lots of different things that we can do in autism, and they're all worth doing. I think that we don't want to lose sight of the fundamental goals of the social communication, repetitive, those core features that mark autism. And we do want to treat the GI symptoms, and we want to treat sleep, and we want to help kids with pressure vests, and those are all important things to do, but it doesn't mean that it's, we also have to focus on how can we really fundamentally address the root cause of the autisms, and how can we fundamentally address therapy that's going to make a huge difference, as well as the incremental difference. So I'm not aware of any therapeutic approaches that fundamentally address that issue. Device approaches. But, if there were, that would be great, because we're definitely seeing devices in other therapeutic areas that are starting to make a real difference. There's devices that cure diabetes. There are a number of different approaches to devices that can really make a substantial difference in disease. I'm not sure what that approach would be in autism.
I see Dee Dee is looking at us very seriously over there. I'll just add to that, the only thing that I've seen is that to help potentially behavioral modifications, the use of sensors, for biofeedback is something that I've seen.
Can we take another question?
I just had a question. Are there any cutting-edge drugs or meds that are used to treat the person or the child or the adult who's greatly affected by autism? And also, which pharmaceutical companies are working in the direction of coming up with meds or drugs to treat autism?
Who wants to take that question?
Well, the ones that I'm aware of that are working in, certainly Novartis in town is working on the development of, or is interested in the development of autism-based drugs. Roche, I know, and its sister arm Genentech both have groups of people involved in that area of neuroscience. But as far as cutting-edge drugs that are available today, as I think was said earlier, there are really only 2 approved drugs that only treat symptoms, not really treat the disease.
I think what's exciting is, as Ellen said earlier, is that we finally have, I think, a sense of hope in this field, that even big pharma like the ones that Peter mentioned are actually interested in at least investigating potential interventions in autism. I think the exciting progress that's happened in the preclinical area and academic research, I think, has brought in those companies into the realm, and that I'm not sure, we can discuss this at another conference. It's finally we're seeing if there might be a market for autism drugs, and big pharma is starting to think about it. They're still at very early stages. Each of them is taking a different approach, using some stem cells, some mouse model approaches. So it will take several years to get us drugs in the market. But the fact that they're involved and they're engaged and they're working with academic investigators, I think is a huge step forward.
Should we?
One last question? One last question, sorry.
Well, all the talk about all of the, is it thousands of genes that are associated with autism, and it's still 2% or 20%, but the first genetic disorder associated with autism was phenylketonuria, PKU, and that clearly had to do with a toxic byproduct of a faulty enzyme affecting the brain. And the brain needs to be the focus of research in autism. And PKU has been treated successfully with a diet that's low in phenylalanine. And I just wonder how many of these other genetic disorders are causing enzyme dysfunction that produce a poisonous byproduct that damages the brain. And the final common pathway in the brain has to do with language learning, and probably the basal ganglia have to do with the repetitive chorioathetoid movement disorder. And so I think when we're talking about developing drugs, the other very scary thing is that all of these drugs have side effects that clearly affect the brain when you have extrapyramidal symptoms and Parkinsonism, and you have to develop carbidopa to counteract, the effect of Thorazine or whatever. But I think the brain needs to be the focus, and how the brain is damaged by any toxic substance, whether it's produced by a genetic substance or something in the environment.
And, thank you very much for that comment. I wish we had more time to talk about each one of those comments you raised, but I think what we try to express today is that there's a lot of enthusiasm. The genetic causes are just being identified. There's going to be some metabolic causes, maybe some milder cases of metabolic diseases that we haven't identified in the past, but some of them might be reversible or treatable. I think what's exciting is that there's now a sense that they're becoming tractable, and hopefully they will translate into clinical treatments in the next several years. Thank you so much, everybody.
Well, thank you, everybody. Is this microphone on? Everybody hear me? I just want to make some closing comments. First of all, on behalf of the fellow board members here, I'd like to thank Dee Dee and her volunteers and staff for putting on, another great conference here. Matt State and Rudy this morning, as well as Sarah, gave us great insights into some of the promises of what we're learning about autism and what we might be able to do with it. And of course, it's always the highlight listening to the ARSs and what they're doing for the families. It's really, I think, one of the most proud things that we have done at the Autism Consortium. As you've heard throughout the day, we at the Autism Consortium are very proud of what we've all achieved. You heard from Lenny Rappaport, Chris Walsh, Maganka Sir, who got up after Paul talked about our transition. We're not only very proud of what we have accomplished, we're very proud of the way we are making what we've done into the DNA of this community. So on a personal note, I'd just like to talk a little bit about Paul and Ann and embarrass them because they knew I wouldn't do this. First of all, I've got to know Ann quite well. Ann is very smart, funny, and very foreboding when she wants something, individual. So I'll come back to that in a minute about Ann and Paul. But let me tell you a little bit about Paul and how we met and why I'm actually president of the Autism Consortium Board. So leadership has been described as a process of social influence in which one person, Paul in this case, can enlist and aid the support of others in the accomplishment of a common task. For example, some understand that a leader simply is somebody whom people follow, or as somebody who guides or directs others, while others define leadership as organizing a group of people to achieve a common goal. Paul redefines leadership. Great leadership is not about doing things right, it's about doing the right thing. That defines Paul. He moves with the speed of thought and a dazzling array of ideas that at first are overwhelming. But when you finally get it, and we all finally got it, it all makes perfect sense. And then you wake up the next day and say, what did I agree to do? And how are we going to get that done? And yet, here we are today. We would not be here today if it wasn't for Paul. We saw his vision, each of us who co-founded this with him, and he understood the steps necessary to get from there to here. He recruited, well, he did some arm twisting, I guess is better put, all of us each into this mission. He is fearless, and he never took no for an answer. As Steve Hyman suggested in one of our first meetings, lionize those who help and take out back and shoot those who don't. We actually do have a large stack of bodies in the back of the building here. But then again, we have an army that stands close with us in the mission that we all have been so passionate about. This is about the power of collective impact, and that's what you've seen here today and the previous conferences. Not one man, not one research paper, not one clinician and her practice, not one lab or not one institution. The kind of breakthrough science and practice needed for the brain requires all of us, in partnership with patients in the community. And there was only one couple capable of making that happen, Ann and Paul Marcus. Thank you very much. So thank you all for coming, and we look forward to see you each and every year as we transition just from autism into neurodevelopment disorders. Thanks again.